Welcome to our webinar, Troubleshooting Immunoprecipitation. I guess most of you are familiar with immunoprecipitations, but I think it makes still sense if we first get a general overview of the different steps of an immunoprecipitation reaction before we start with the troubleshooting. So if you do an immunoprecipitation, you want to isolate your protein of interest from cell lysate using antibodies coupled to a solid matrix. So if you start your IP, you have to choose an antibody. And this first step is probably already the most important step in your IP. Because if your antibody is not working for IP, your whole experiment won't work. This antibody you then couple to a matrix. Mostly beads are used like agarose or magnetic beads. After choosing the antibody, you have to prepare your cell lysate. Cell lysates can be prepared from many organisms like eukaryotic cells, yeast, plants, so it depends which organism you're working on. To start your IP, you then add your lysate to the antibody coupled to beads. After incubation, you wash away all unspecific bound proteins. And finally, you can elute your protein of interest from the beads. This eluted protein you can then use for all kinds of downstream applications. So the principle of an immunoprecipitation reaction is very simple and straightforward. However, all of you who already did an IP know that there can be many pitfalls during such an IP. So now let's start to have a closer look at the single steps of an IP. As I already mentioned, the first and probably most important step is the choice of the antibody. And remember, not all antibodies are working with IP. Frequent problems that might occur is that your antibody binds unspecific to many proteins. Unspecific binding will result in a high background in your IP. Or your antibody does not bind to your antigen or only weakly. Then you won't get any IP. And please remember also that antibodies which work really good in vessel plot might not work for immunoprecipitation because these antibodies only recognize denatured proteins. To avoid these problems, you can use antibodies that are validated in IP, antibodies which are published in IP experiments, which are recommended, or which are certified. But if no good antibodies are available against your protein of interest, think about fusing your protein to a protein tag. Good antibodies are available against protein tags like HA, MUC, FLAG, or GFP. After choosing an antibody, you have to choose a matrix. Often protein A or G beads are used, since many antibodies are bound by protein A or G. Using protein A or G beads, you are very flexible. But if you want to elute your protein, you have to remember that you not only elute the protein but also the antibody. And depending on your downstream applications, the heavy and light chains of your antibody can be disturbing. To reduce heavy and light chain contaminations in your downstream application, you can cross-link your antibody to protein A or G beads. Or you can couple your antibody directly to activated beads. Using this system, you are still very flexible, but it is more time consuming and needs more validation. A third option is to use beads that are ready to use. You just take them out of the fridge, wash the beads, and start your IP. This system is very fast since the antibody is cross-linked to the beads, you again have reduced or even no heavy and light chain contaminations in your downstream application. The disadvantage of ready-to-use beads is the limited choice of antigens. So ready-to-use beads are probably not available against all proteins you are interested in. After choosing the antibody and the matrix, you need to lyse your cells. During lysis, you want to solubilize your protein. Ideally, your protein is kept in its native conformation, and for sure you don't want protein degradation during cell lysis. And if you're interested in post-translational modification, you also don't want to lose these post-translational modifications during lysis. At least for eukaryotic cells, mainly two buffers are used, which solubilize nearly all proteins. There are more difficult proteins like membrane proteins, for example, but discussing solubilization and IP of membrane proteins would definitely extend the time in this webinar. But as I said, for many buffers, for many proteins, you need mainly two buffers. The first buffer is a non-denaturing buffer. It contains triton or NP40. This buffer is very suitable if you want to do co-IPs, since it does not denature your protein. But you might have problems to solubilize some proteins like nuclear proteins. To solubilize these proteins, you can use repair buffer. However, repair buffer is slightly denaturing and you might have problems with co-IPs. And again, please don't forget to add protease inhibitor or other inhibitors like phosphatase inhibitors if you are interested in such modifications. After preparation of the lysate and choice of the antibody, you can start your IP by adding the antibody to your lysate. As mentioned before, you need a good antibody to catch your protein from the lysate, otherwise your IP won't be successful. But you also might have to optimize several other parameters during your IP. So you can play around with the salt and detergent concentration, and you can also try to adjust time and volume of your IP reaction. The same conditions you can optimize during the washing step. So you can again optimize salt and detergent concentrations and time and volume. Elution from the beads is often done with denaturing SDS sample buffer. But if you need your protein in a non-denatured form, you can try to elute with glycine pH 2.5. But keep in mind that you immediately neutralize your protein after a glycine elution. Afterwards, it will be also denatured. Depending on your downstream application, it might be also an option for you to use your protein bound to the beads and avoid elution. But after finishing your IP experiment, you normally check by Western blot or Coomassie whether your IP was successful. If it was successful and you see what you expect, it's fine. But often two problems occur. The first problem is that you have only a weak or no pull down of your protein of interest. If you haven't already done it, the first thing you should do, check by Western blot whether your protein is soluble in your extract. Maybe it is still in the pellet and then your IP can't work. You should also check whether your antibody binds to protein A or G beads, because some antibodies are only weakly bound by protein A or G. Prolonging the IP reaction might also help you to get better results, especially if your extract is very diluted and your antigen concentration is very low. Another problem might be that you have really high background in your bound lane. So to reduce background, you can try to prolong the washing steps. So go up to even 5 minutes if your protein or protein complex is stable. You can increase the stringency during the washing steps. You can increase the sample volume during your IP. This might also help. You can optimize the detergent concentration, and in our experience, if you transfer the beads to a new tube after the first wash, all proteins which are bound to the tube will stay in your tube, and you will transfer only the proteins that are bound to the beads, and this might also reduce your background. So during the last slides, we discussed many parameters which can influence the success of your IP. And all these parameters you have to optimize again and again for every new antibody. But as I already mentioned, good antibodies are available against tag proteins like MYC, HA, FLAG, or GFP. And if it is possible for your application, you might fuse your protein of interest to protein tag. Then you know at least that your IP can work in principle and that the antibody is not the problem if your IP is not working. And you can concentrate then on all other parameters for optimization. And a really good antibody against the tag protein is the GFP-Trap. The GFP-Trap binds with very high affinity to your GFP fusion protein. And using GFP as a tag has also the advantage that you can easily combine microscopy and biochemistry. But now let's have a look. What is the GFP-Trap? So conventional antibodies have a size of about 150 kilodalton. Camelids have, besides the conventional antibodies, also so-called heavy chain only antibodies. And the antigen binding region of these heavy chain only antibodies is the smallest naturally occurring antigen binding fragment. In the case of the GFP-Trap, such a small fragment directed against GFP is covalently coupled to matrices like agarose beads, and this resides then in the so-called GFP-Trap. And especially the strong binding of the GFP-Trap to GFP is a big advantage in optimizing your IP reaction. The GFP-Trap is already or was already used in thousands of IP. It's published in more than 500 publications. The beads are ready to use, so again, just take them out of the fridge, wash them and use them. And GFP-Trap is available coupled to different matrices like agarose beads or magnetic beads or even 96-well plates if you want to do high throughput analysis. The GFP-Trap is also working in all standard lysis buffers. But even if you have a special buffer that is you need to use for your lysate, the GFP-Trap will probably work. And the GFP-Trap recognizes the native form of your GFP protein, so meaning that your fusion protein is probably also in its native conformation. In the following experiment, GFP was immunoprecipitated from cell lysates using the GFP-Trap. Pull-down efficiency was checked after 1 minute, 5 minutes, 10 minutes, and 30 minutes by Western blot. You see that after 10 minutes, only a minor band is visible in the flow-through lane, and most GFP was already precipitated from the extract. And after 30 minutes, all GFP was immunoprecipitated and is in the bound lane, and no GFP is anymore visible in the flow-through lane. So this shows that the GFP-Trap, the pull-down efficiency of the GFP-Trap is really good, and that your IP can be very fast. To reduce your background in IP, you might have to wash with really high stringency. Using the GFP-Trap, you can increase the salt concentration in your wash buffer up to 2 molar NaCl, or you can even wash with 8 molar urea. Even during such harsh washing conditions, the GFP-Trap will bind to GFP. So elution from the GFP-Trap can be done with SDS sample buffer, or you can elute with glycine pH 2.5, or you can also use your beads directly in downstream applications. Finally, I will show you a couple of possible downstream applications using the GFP-Trap. So most of you probably do a Coomassie or Western blot after immunoprecipitation. In this experiment, a pull-down of GFP with the GFP-Trap was compared to an IP using conventional antibodies. As you see by Coomassie and also by Western blot, the GFP-Trap pulled out the GFP very efficiently, and no protein is left in the flow-through. With conventional antibodies, GFP was also pulled out, but with less efficiency, and you see that in the Coomassie, and you also might see it in Western blot, that you still have heavy and light chain contaminations in your Coomassie and Western blot. And this might really be disturbing for you. Especially the harsh washing conditions can be also an advantage if you want to do mass spec, because you need really clean IPs for mass spec. And this is also very well suitable if you use the GFP-Trap. You can also do enzyme activity measurements either by using the beads directly or by eluting it with glycine. And it is also possible to do chip and rib analysis. So to summarize the webinar, we showed you that immunoprecipitation is often not trivial, but don't be scared. And the success of immunoprecipitation is influenced by many parameters, especially the choice of the antibody. So that you can focus on your experiments, you might use established state-of-the-art tools like TAG antibody if it is possible. So now I would like to thank you for your attention, and if you have problems or you need information, please just write us an email and we can contact you.